Hello. The first video I'd like to put up is about prokaryotic cell division. First of all, if you don't know what a prokaryote is, definitely check that out before watching this video. That's much more important. Alright, so like I said, this is about cell division and the cell cycle. So for prokaryote, it's very, very simple. We'll see that eukaryotic cell division is much more complex in a moment here. So first, this dark line inside here is the cell wall. Right inside here, that's the plasma membrane. DNA is right here. And then, I don't know if you can see up here, that's the notch. And when we say that the first step has to do with notch formation and DNA attachment, the attachment that we're talking about is to that notch. Okay, so that's it for that step. And then moving on to step number 2. In step number 2, you essentially just have duplication of DNA and a new copy of DNA. You can kind of see the one DNA here and then the second molecule down here. The new copy, the copy that was just made, is still attached to the notch, but the old copy has detached. And pretty much the only thing that happens in that step number 2 is that duplication of DNA. Moving on to number 3, what is the dotted line in here that you see is the septum, and the septum is essentially the place where the new cell wall and plasma membrane forms. And in this stage, what happens is, first of all, these two DNA molecules that you see have detached from the notch, and they're both just free floating on either side of the septum. And let's see here. Yeah, so the septum begins to form, and after that, of course, the cells are going to divide. And this stage, if you want to think of it in terms of words, is just cell elongation, meaning the cell, of course, gets longer, and detachment of the DNA. Alright, now the last stage we see here is, of course, the formation of two identical sister cells from the one bigger mother cell above. Those cells are identical genetically, and one thing that Dr. Said wanted to stress was that this stage can occur in about 10 minutes. I'm sorry, all 4 of these stages together can occur in only 10 minutes. Prokaryotes, as hopefully you know, are very simple compared to eukaryotes. So it's not surprising that they can have such a low generation time. And as a result of that, it's necessary for the cell, if it's going to do it that quickly. I think E. coli has a very low generation time. Don't quote me on that, though. If the cell is going to reproduce itself in only 10 minutes flat, it's going to need to carry out those steps right on top of each other. In other words, it's going to have to start 3 before it actually finishes 2, and start going, like maybe it'll start forming the septum, but then sneak the DNA in there before it happens, that kind of thing. Okay, and another thing he wanted to say was those prokaryotic cells, by different means that we didn't go over in that short summation, are those daughter cells will be roughly equal in terms of the number of plasmids they have, in terms of the number of pili they have, and that entire process of prokaryotic cell division is called binary fission. Alright, I think that's it. A few more minor points. We said in step number 3 right here, the septum is formed. The peptidoglycan, which is a major component of the bacterial cell wall, will be formed actually in the cytoplasm and then shipped to wherever it needs to go. So that's how it'll arrive at the septum. And another thing I should say is I stressed earlier that the prokaryotic cell division can occur very, very quickly. Like in 10 or 12 minutes would be close to the fastest. But it can also take place over a much longer period of time. And no matter how long it takes, that process of cell division is called, well, the amount of time that it takes is called generation time. Alright. Yep, I think that's it. Another thing he mentioned in the lecture was that the peptidoglycan, which is, like I just said, the major component in the bacterial cell wall, penicillin actually prevents the formation of new peptidoglycan. So cells that are actively dividing are very weak against penicillin. So that's another thing to consider. Alright. The next video I put up, I think, will be about eukaryotic cell division. Thank you.